Good morning, everyone, and welcome to the Medical School Seminar Series. I'm Antonio Tricoli, and I will be chairing the symposium today. I would like to start by paying my respect to the First Nation custodian of the country throughout New South Wales, and in particular, the Gadigal people of the Eora Nation, the traditional owners of the beautiful land on which the University of Sydney is built. I pay my respect to their elders past, present, and emerging. And today we have the luck to have with us Kevin. Kevin is a group leader at the University of California in Riverside, and he obtained his PhD in chemical and biological engineering at Drexel University. Upon that, he was at Imperial College London as a postdoctoral White Baker Fellow. And then he actually has been visiting quite a few universities internationally, such as UPenn and KAIST in Korea. And he joined the Global Viral and the LBL Helms Genome Institute, working on single-cell genomics, metagenomics, and bias-free sequencing. And so today he will be talking about something very exciting, especially now that we consider that biomedical diagnostic is so important in our life. And this is about molecular profiling using nanopore device, what information is obtainable, and then really curious to hear more about this. Before we start, let me speak about the housekeeping. We will have a seminar for 40 minutes, and there will be after 15 minutes of questions. Please mute your microphone and keep your camera off during the seminar so we can actually maximize the use of the bandwidth. And upon the seminar, we'll open actually the presentation for question. So please raise your hand or send me a message in the chat, and they will let each one of you ask a question after each other to keep a little bit of order. So thank you for that, and I would like to take no more of your time and let Kevin start.
Thanks, Antonio. Can everyone hear me okay? And see my screen? Okay. Like I said, my name is Kevin Freedman. I'm an assistant professor in bioengineering. My lab focuses on single molecule techniques. Probably the biggest portion of my lab focuses on nanopores. And so what I hope to present to you all today is what information can we achieve or measure using these devices. Okay, so the very first question you might have is, where is Riverside? So we are east of Los Angeles. San Diego is down here, it's cut off from the map, but Palm Springs, you may be familiar with some of these. San Bernardino, the birthplace of McDonald's, and Riverside is right here. Okay, so what is nanopore sensing and how did it get founded, right? So the very first, what we consider as like the birth of nanopore sensing is this PNAS paper, which did a very simple experiment. They took single-stranded DNA and passed it through a protein pore. And at the very last paragraph of this paper, they make this future speculative guess that they might be able to use this for sequencing DNA. And so this is kind of the fuel behind innovation in this field, is this idea that we can maybe one day sequence DNA using nanopore technology. Okay, and so this paper, like I said, was using a protein pore. This is a pore called alpha hemolysin. It's a heptamer, so it has 7 different subunits, and it has a very reproducible inner diameter. And so the DNA flows through this open portion. Okay. And so, like I said, the fuel behind innovation here is the promise of DNA sequencing or genome sequencing. And so this is a plot showing the cost of sequencing a human genome as a function of time. And so in the early 2000s, we were mostly using Sanger sequencing. And so, it's declining mostly due to a reduction in reagents and, somewhat new technology allows us to use lower reagents. But it really takes a really, sharp dip when there's new technology that enters the market. And this is next generation DNA sequencing that enters around 2007. Most notable is Illumina, which is based out of San Diego. And so we see this huge drop in the cost of a genome. Some people may say that Illumina actually has achieved the $1,000 genome. It's kind of debated. But nevertheless, we've seen this huge drop. On this graph, it levels off at about 5,000. Okay, so how can we get it down to 1,000 or even lower? And so that's where we have to, again, inject a new type of technology to get us there. And so we're hoping that nanopore technology can do that. And even though this seems like a very small decrease, we're hoping that this, tenfold drop, maybe 5,000 to 500, will be able to get DNA sequencing into our local doctor's office. So if a doctor can do a biopsy, see if there's any DNA mutations in a tissue, and it'll just improve healthcare, make personalized medicine possible. Okay. And just to kind of show that the 4 bases of DNA can indeed be discriminated, there is a paper in Nature Nanotechnology in 2009, which showed, again, passing these individual nucleotides through alpha hemolysin. We see that there's this, 4 peaks corresponding to the 4 bases in DNA. Okay, so how can we take this original idea of using alpha hemolysin to sense molecules and make a solid state version? This ultimately is what my group focuses on, is solid state nanopores. And so we kind of take this, we call it biomimetic because we're trying to achieve the same properties of that original pore, namely that it's non-gating, so it's open all the time. And so we can take solid state materials and use electron beams and ion beams to drill individual pores into these membranes. Okay, and so these are roughly the same diameter pore here. And so one area that we can try to use this technology is in commercialization. And so there's actually an Oxford Nanopore device that's floating above us in the International Space Station. The other area is doing biophysics. So can we take these molecules and learn something about how they behave, especially in non-equilibrium situations and environments? Okay, so taking a native protein, for example, and looking how it could misfold, what the structure looks like. And typically whenever there's some change in folding, there's either a loss in function, or even worse, there's some pathological state that may present itself. And so here you can see the MinION. This is Oxford Nanopore's device. And so it's just a USB drive. You pipette your sample and it outputs a DNA sequence. Okay, so what is the mechanism of nanopore sensing and DNA sequencing? The basic idea is that you have a single pore, and we put the pore in a salt solution. In often cases, this is KCl. So we have potassium and chloride, and we apply a voltage across this membrane. And so when we do that, we get charge separation. Current flows through the pore. When something occludes the pore, like DNA, we get a reduction in the number of ions that translocate. Okay, so this is a relatively simple process. This is just essentially electrophoresis of ions and DNA. DNA, of course, is negative, so it gets attracted to the positive electrode. In reality, there's a lot more forces involved. Oftentimes, the membrane is made out of silicon nitride or glass, and these are both negatively charged. And so you get accumulation of counter ions, in this case, potassium. And so this volume of ions is net positive. And so this also is going to migrate in an electric field. And so we have electrophoretic force, in this case, going in the down direction. Oftentimes there's an excess of DNA in the top chamber. And so we have some small diffusive force, even though the concentration gradient is very, very small. And then this can be very dominant. So the electroosmotic flow can provide a convective force in the opposing direction. And then one of the first topics that I'm going to talk about today is how can we apply and modulate these forces using pressure as well. Okay, so this is the basic experimental setup. We have something that applies a voltage, and then we have a very sensitive ammeter. So this measures the current passing through the pore. Okay, and we get this ammeter is sensitive to femtoamps. So it's very small currents that flow through the system. Okay, so my focus is on the technology. I'm interested in all forms of biology and physics. Okay, so even though the same approach is used, we like to apply this to all sorts of different systems. I'm really interested in the nanoscience aspect, so transport properties and the sensing physics. But we can also track disease progression using single molecules. We can try to sequence proteins, which is something that we're trying to do in the lab now. Epigenetics, we can look at viral assembly. Viruses tend to be on the 100 nanometer scale. And so we can design pores that translocate viruses. We can look at protein-protein interactions, detecting disease biomarkers and doing diagnostic sensing, DNA sensing, and either lipid-based or synthetic characterization of nanoparticles. So what are the benefits? Okay, so there's lots of different other single molecule techniques out there. There's AFM, atomic force microscopy, magnetic tweezers, optical tweezers. However, you know, and these all have their benefits. The benefits of nanopores, however, is that these are very high throughput, as opposed to these ones where essentially the sensor is moving. In the case of AFM, the tip has to scan across the surface. In our case, the molecule is what's moving. And so the molecules can be transported through this pore very quickly, and the sensor stays stationary. Okay, so that's one of the properties that we like to mention, is that how high throughput the technology is. It's also label free, so we don't have to use fluorescence to label these molecules in order to detect them. They can also be mass produced pretty cheap. And not only sensing, but we can also apply perturbation forces. So we can look at kinetics, and we're going to look at the very end of my talk. I'll talk about how electric fields can induce certain kinetics in proteins. The nanopore also can be used to introduce denaturing gradients. So we can apply different chemicals inside these two chambers. So we have a top chamber and a lower chamber. We can change the fluid conditions as well. So I really enjoy nanofabrication, so I'm going to talk a little bit about nanofabrication before getting into its use as a sensor. And so solid-state nanopores are typically in a chip format. So there's a silicon support here. This is my tweezers here holding the chip. And in the very center, there's an open etched window. We call it a window. It is transparent to optical light, and it's about 50 microns by 50 microns. And once we have this open, free suspended silicon nitride window, we then put it inside a transmission electron microscope and focus the beam until a pore is formed. This is the most common way about 10 years ago. More recently, a lot of people have been using a technique called controlled breakdown, where you just apply a very high voltage until a pore is formed in this material. But this is still used quite often today. Okay, so we have fine control over the diameter by modulating this beam intensity. We can also use a focused ion beam. So electrons are a lot smaller, and so using an electron beam provides much smaller pores. So we can go down to 2 nanometers with an electron beam. With a focused ion beam, usually the smallest is around 20 or 30 nanometers. One of the things that you can do, however, is using a scanning electron microscope, so a different instrument. You transfer it into SEM, and you scan the surface, and you can actually get the pore to shrink. And so this was some early work that we did showing that you can control the pore diameter post-fabrication. And so this is the very same pore, just at different time points. And so it shrinks and can be tailored to the right size. We can also make 2D materials and use those as membranes as well. the most famous of which is graphene. So this is a one atom thick membrane. And you can see here, as we change the drilling time, this is the time that the electron beam is focused to a spot, we can change the pore diameter, okay? And so we have really nice control over the pore diameter in this case as well. So that's the fabrication side. And then once we form these pores, we pass molecules through it. The very last technology or type of nanopore that I'm going to talk about is these quartz nanopipettes. These you don't need any electron microscope. This is a benchtop unit, and we put in these glass capillaries and mechanically heat and pull them until we get nanoscale tips. And so these are, after fabricating them, we can image them, and you can see the internal diameters. The smallest one we've been able to pull is around 5 nanometers. So we have pretty good control, and we can go all the way up to micron scale. Okay, so now getting into the experimental stuff. One of the things that we wanted to do with these pores is see if we can capture the same cell or same molecule more than one time. Okay, so in order to do something like this, you have to apply a different force. And so we were looking towards using pressure to apply this secondary force. And so we used this very large pore initially, and we used 2 different types of bacteria. One is a rod-shaped and one is a sphere. Okay, and we're using this piezoelectric pressure controller, which can apply positive pressure and a vacuum. Okay, so we have nice control. We can either push fluid out of the pore or pull fluid into the pore. So one of the basic questions, and a few of the basic questions that we had was, what pressures are needed to overcome electrophoresis? Because we still have to apply voltage. That's something that all experiments have to apply. What is the capture volume? So how far away can a cell be before it gets sucked through this tip? Can we recapture cells or molecules, and what are the limits of recapture, especially in terms of size? So if we make the pore smaller, can we still recapture things? And the answer to that, kind of to skip ahead, is that it becomes much more challenging as we make the pore smaller. How does pressure affect the signal? Okay, so there's been a lot of, we aren't the first people to apply pressure to a nanopore. It's been done before. However, in all these papers, the only property that they look at is how long it takes for a molecule to pass through the pore. So they're interested in slowing down the translocation rate of these molecules. And so we had a lot of other basic questions to explore. And so in order to sort of visualize this process, we first looked at the tracking 1 micron beads. And so these are fluorescent images of 1 micron beads. And I drew these rectangles here to indicate where the nanopipette is located. Okay, so we have this nanopipette, and we bias the inside towards a negative pressure. So there's a negative suction going into this pore, and you can see that these beads are traveling through the tip. The particles out here, they still experience a force. However, it misses the pore and kind of just gets pushed away up the taper of the pore. Okay, so this looks very similar to our COMSOL simulation. So these are finite element models. And so you can see how they resemble each other. So most of the captured beads are occurring sort of in the front of the pipette. So all the beads that are out here get sucked through the pore. The other question we had is, naturally there's going to be electroosmotic flow. And so that's what's showing here is there's a very weak electroosmotic flow when there's a positive voltage bias inside the pore. So there's a positive voltage bias. And so that's going to drive the potassium out of the pore. However, by applying a negative pressure inside, this completely reverses the flow, and we can then have pressure be the dominant force. Okay, and so we can also, measure the beads going through the pore. So this is a integration of optical fluorescence at the tip, and so we can see these beads. Each one of these spikes represents a bead going through the pore. Okay, so what effects does pressure have on the current signature? And so going back to our original model, the pore is either in an open state or a blocked state. And so what we see here are these current reductions when in this case, these are beads, when the bead goes through the pore. And so the first thing that we noticed was that there's a drastic reduction in the magnitude of this current drop. So we have a current drop that looks like this, and then it gets smaller and smaller as we apply the pressure. And this is something that hasn't been reported before in literature. And so you can see this 1 over current drop relationship on both sides of the pressure curve. So whether we apply a vacuum or a positive pressure, in both cases, it reduces the current drop of these beads. It also, as we apply more and more pressure, it also makes the time spent inside the pore go down. Okay, so this is dwell time. This is the time spent inside the pore, or essentially the full width half max of this event. Okay, so what properties can we measure from molecules or particles? One of the things that we can measure is excluded volume. So how can we convert this delta I, essentially the current drop, to volume? And so that is done using this equation. In previous published literature, pressure hasn't been included. So we had to derive a new equation to take into account pressure. And so the nice thing about this equation is that if the delta P, or the pressure bias, is 0, you just regain the original excluded volume equation. So the original equation is just sigma V in the denominator. And so you can put this 1 plus phi delta P either in the bottom or in the top here. And so what we found is that when we use this new equation for excluded volume, before we were getting all different types of volumes for 1 micron beads. However, after using this equation, all of them kind of collapsed onto each other, and we got the true excluded volume, which is about 0.53 microns cubed. And so this is the true excluded volume for a 1 micron bead. So, like I said, we originally started to use pressure to explore the ability to sense a cell and then bring it back to the sensor. So somehow sense a cell and then sense it again. And so by applying a sinusoidal pressure wave, we can do that. So this is the current on top. This is the initial event for 1 micron bead. The pressure then switches. There's a little transient here wherever the pressure is 0. And then we capture the bead again. So we get these 2 measurements of the same exact bead. And this is also possible using that same mixed microbial population. So we have rod and sphere microbial cells. And so we can do the same thing with that sample. In order to figure out how successful this pressure-based recapture system is, we wanted to employ a closed loop type of recapture, as opposed to using a sinusoidal pressure wave. This is kind of what we call the open loop configuration, because we can't control when this initial event happens. So if an event happens here or here, we have no control over that. And so, and that's going to impact when it gets recaptured over on this side. So to fix that, we applied a real-time event sensing algorithm. And so it detects this initial event, waits a prescribed amount of time, So this is called the switch time. So this is the time that we wait and then switch the pressure. And then this is the recapturing of that same bead. So this is the release time. We call it the release time because the bead is going into the nanopipette and then back out the pipette. And so one of the things that we found is that we get 100% recapture probability using pressure. And one of the reasons why flow is so good at recapturing is because this is all laminar flow. And in most cases, this is fully reversible. So if a bead goes into the pipette, we can reverse the flow and have that same bead come out at a later time. And so we're getting this really high efficiency for recapturing. Not only that, but we can recapture into the seconds range. In most cases, recapture technology, you have to reverse whatever force within a few milliseconds in order to get it back again. And we've extended that here. We can wait a few seconds and still get it back again. Okay, so we looked at event properties and how we can recapture these micron-sized particles. What does the recapture really tell us? So we want to assign some sort of meaningful information about what this recapture time means, mainly because we saw differences in the recapture kinetics for these two different populations. So for 1 micron spheres, if we wait longer to switch the pressure, this results in a longer release time. So we get this linear relationship. And for the microbial population, we see these two independent linear relationships. And so what we think is happening is that either due to mass or charge or some mass over charge relationship, we're able to discriminate between these two cell types. And so we wanted to explore this, and ultimately we believe that what we're sensing in this case is mass. Of course, mass and charge is what we're sensing, but in the case where charge is constant, a difference in recapturing is going to be due to mass. In the case where mass is constant, any difference in recapturing is going to be due to charge. And so we've computationally modeled what is happening in our experiments. So we use the same nanopipette geometry, the same pressures, the same voltage, and there is a mass-dependent acceleration that happens whenever a cell changes course. And so we believe that this discrimination property is due to one cell type being more massive than the other cell type. We also calculated the magnitude of the acceleration that is experienced in this system and compared it to something that is a known inertial mass sensor, which is a QCM crystal. And so this is known to have acceleration forces on the surface that go up to about 1 million times the Earth's gravitational field. And so we calculated that the magnitude of the G forces is around 250K for the pressures that we were using. So there's definitely going to be some inertial effects going on here that correspond to mass. One of the other things that we wanted to explore is why are nanopipettes so good at accelerating fluid, and ultimately we found out that it's really the taper geometry that plays the most important role in mass discrimination. So we modeled this channel which has the same opening diameter as the nanopipette. And in order to get any sort of mass sensing, we have to switch the pressure back and forth at 10,000 hertz. So we either have to switch the pressure very quickly, or we can make the acceleration forces maximized by using this taper geometry. And you can see that the magnitude of the velocity is around 2000 in this case, and only about 80 in this very fast frequency switching. Okay, so we already kind of established that we can look at excluded volume. and recapture things. And one of the things that I was interested in looking at is, can we deform a cell and even potentially rupture aggregates of cells? And this is something that we can uniquely look at using recapture technology. So if we push something through the pore and it has a certain property, and then we remeasure it again at a later time point, any differences there are going to be due to the pore itself. Okay, so here we have recapturing, we have 6 different recapture events of the same bead, or sorry, same cell aggregate. And so you can see that the event signature is indicated 1, 2, 3, 4, 5, 6. And I just took passages of 2, 4, and 6 here. And you can see that the event changes over time. And what we think is happening is that there's a rod-shaped bacteria adhered to a spherical bacteria, and that these 2 cells are being eventually, cell adhesion is rupturing. Okay, and so we saw this multiple times. We inferred different cell populations or cell aggregates to each one. In this case, we think that the product is, by the magnitude of these current drops, that these are probably the same type of cell. So we speculate that this is an aggregate of 3 of the spherical cells, and we're breaking it apart through the passage of 12 different recaptures. So clearly there's some force being exerted onto these cell aggregates, but what about a single cell? So these are bacteria. So there's 3 different there's 3 different membranes here. We have an outer membrane, a cell wall, and a lipid membrane. And one thing that's also present is there's an outward pressure here that corresponds to the turgor pressure of a bacterial cell. So we explored many different possibilities that could be happening in our system. For example, we could be experiencing pressure squeezing the cell, making the entire cell body shrink. That might be what happens if the pressure is greater than the turgor pressure. In most cases, what we think is happening is that the shear forces are actually stretching the cell body. And so this hydrodynamic stretching is what we think is happening. And we see that here by a change in the excluded volume. So the same cell is going to have the same volume. And so to account for the discrepancy in volumes, we introduced this additional factor, which is the shape factor. So a shape factor of 1 means that it's the same cell and no deformation. A shape factor that's smaller means that the cell body was deformed in some way by the exerting of the pressure. So for this cell type, we are seeing a change here. And so there is some deformation of the cell body happening. Okay, how can we take this a bit to smaller scale, nanoscale entities? And so one of the systems that we've worked on quite a bit with a collaborator is characterizing nanoliposomes. And so there's 2 different nanoliposomes that we've worked with. One is synthetic and one is red blood cell derived. The benefit of using red blood cells is that if you take red blood cells from a patient and put drug in them, there won't be an immune response to those lipids. And so as a result of that, that nanoliposome will circulate within the bloodstream for longer without being filtered out. And so the effect of the drug should be elevated. And so we wanted to characterize these red blood cells not only for concentration, but also looking at particle size, deformation properties, and also trying to figure out if we can look at the internal contents of the nanoliposome. Okay, so these red blood cell derived nanoliposomes are actually really hard to measure the concentration of. And so one good technique of using, or the advantage of using nanopipettes, is that we can measure concentration without changing the particles at all. We don't have to add fluorescent dye, and these are nanoscale, so we can't image them. And so if we get particles per second from our measurement system, and we know what the flow rate through the pipette is, this is inverse flow rate, then we can get particles per microliter. And so this is how we calculate concentration for these nanoliposomes. So using empty nanoliposomes and drug-filled nanoliposomes, we found that the excluded volume is nearly constant between these two samples. However, we did find translocation kinetic information that is different between these two samples. The first one is the dwell time. This is how long it takes for the particle to pass through the pore. And one thing to keep make note of is that when a nanoparticle enters the pore, it's going to be counter the electrophoretic force. And when it goes back out the pore, it's going to go with the electrophoretic force. So we do expect there to be a difference between positive pressure and negative pressure. Here you can see that at negative pressure, so corresponding to this situation, it tends to be higher compared to the positive pressure in both samples. Not only that, but the individual samples themselves also have different kinetics. We saw this in dwell time as well as the recapturing time as well. So the time it takes for a nanoliposome to go in and then back out also changes between empty and drug-filled nanoliposomes. Okay, so going down even further, so we looked at nanoliposomes, which are around 100 nanometers. Can we look at pressure effects sub 100 nanometers? Okay, so we fabricated two different pore sizes. This one is 10 nanometers, and this one is 30 nanometers. So we don't, at these small pore sizes, the hydrodynamic resistance, which measures how easy it is for fluid to be pushed through opening, really skyrockets as we reduce the pore diameter. And so we're not able to switch the direction in which a molecule can go. So we can't do any recapturing at this scale. We still get pressure-induced effects, and that's what we wanted to study a bit more. And so what we have here is the net velocity, which is equal to the electrophoretic drift velocity. So this is going to depend on the electric field and the distribution of the electric field in the system, in combination with any pressure or electroosmotic flow induced convection. And so what we're showing here is essentially a reduction or a change in shape of the capture radius. So if you have a DNA molecule outside of this white circle, it's basically going to be diffusing around and not be captured by the nanopore. Once it crosses this white line, it will eventually go through the pore. Okay, and so we see a change in the essentially the capture volume of the system as a function of pressure. And so just like the beads at the micron scale, we also saw pressure-dependent reductions in the current drop. So this is a schematic of the pore being in the open state versus a single DNA being inside the pore versus two DNA strands of the same molecule going into the pore. And so in both cases, we see a reduction in the current drop that corresponds to these states of the pore. So this is all done with about a 10 nanometer pore. We see this, these kinetics, essentially a reduction in current drop. If we make the pore even a little bit bigger, so if we go up to 30 nanometers, the hydrodynamic resistance decreases enough that we can actually stop the flow of DNA. And so we call this DNA gating because we're essentially turning off the events that occur, which are originally there because of electrophoresis, but we're basically pushing enough fluid that it blocks electrophoretic transport of DNA. And so this is what the current trace looks like. So we have individual DNA molecules going through the pore, and then it stops. This is during a positive pressure where the DNA events stop. And then when we remove that positive pressure, the DNA events go through the pore again. Okay, I'll probably try to go a little bit fast because I'm running out of time, but so we already saw that pressure decreases signal to noise. So one question we had in terms of sensing is how can we increase it? And so one thing that we kind of stumbled upon was that if we use asymmetric salt conditions, meaning we put 1 molar KCl inside the pipette and 4 molar KCl outside the pipette, that this produces very good signal to noise. The only difference here is that this actually produces an increase in current. So when DNA enters the pore, the current actually increases further. And so at the very end, if there's time, maybe I'll get into that, why that happens. But these are slightly different types of events. And so we wanted to confirm that there is a benefit to using this asymmetric salt condition. So we tried using low salt, which also produces these current increases, high salt, which produces current decreases when the DNA enters the pore, and then the asymmetric salt. And so just to summarize, we found that the asymmetric salt yields very distinct populations corresponding to the different folding states of DNA. And so these populations aren't as evident in these other conditions. Okay, so these are what the current signatures look like for the different folding states. So linear DNA looks like these events. Partially folded, where the DNA kind of doubles back on itself, looks like these events. And then the fully folded, meaning 2 strands of DNA are always in the pore, look like these events. And so we've looked at different ways to classify these different event types, one of which is using machine learning. So using support vector machines, we can classify the events based on the degree of current drop and the duration of each event, and look at how this changes as a function of voltage. Another technique that we kind of invented was this principal folding component classification technique. So by simply doing a geometric rearrangement of the data, we can essentially rotate the data to be along this principal axis. Okay, and by doing that, we can put the data into one dimension and do fitting to figure out what is the DNA folding states of the DNA. And so these match up pretty well with the support vector machine classification as well. And this shows the signal to noise comparison between all 3 conditions. The asymmetric salt ones are at the very top. So we do indeed get the most signal to noise using this condition. And this is, there's lots of different applications for DNA sensing. We're interested in using DNA for using it as a carrier. So if we have a long strand of DNA, we can put proteins on the strand and sense them either for diagnostic purposes or looking at Cas9 binding or transcription factor binding. And some people are also looking to using DNA for data storage. So putting structures on the DNA and reading out the bits of information and using DNA as essentially a hard drive. So we looked at this asymmetric salt condition. We also wanted to look at Cas9 binding to see if we can resolve the protein being bound to the DNA strand. And indeed we could. So we have DNA, Cas9. This is the guide RNA, which tells Cas9 where to bind to the DNA. And we get this event structure, which is the DNA with Cas9 also present in the middle of the strand. Okay, and so we're able to detect Cas9 on this DNA strand. And so we're looking at using this to look at off-target Cas9 binding, looking at DNA domain detection, or even virus variant detection. So if we can use the guide RNA to probe these DNA templates to answer the question of, does this sequence exist in my sample? Okay, so we already looked at spherical soft particles and long homogeneously charged molecules. A very different type of animal is these small heterogeneously charged molecules, which are protein. And these are going to differ quite significantly, mostly in terms of its interactions with the wall of the pore, as well as what happens inside the electric field. Okay, so this was some very early work.
But just a note that we're running a bit out of time for questions. So just that you know, you have about 10 minutes left for questions. So keep this in mind.
Okay, yeah, I'm almost done. So this was some early work where we looked at the impact of voltage on the structure of the protein. And so this was using silicon nitride pores. And what we found is that there is a single molecule electric field induced unfolding that happens due to the positive and negative amino acids being pulled in opposite directions. And so we looked at this using 3 different proteins, a wild type and 2 mutant proteins. And we found that the stability of the protein is directly related to traditional urea-based denaturing experiments. So there's a one-to-one relationship that is protein and pore specific, but still gives us the same information. And then we used this again recently to look at Cas9 and look at the voltage dependence. And indeed, we found that it's going to be pH sensitive, but we're really surprised to find out that the maximum unfolding happens at the protein's isoelectric point. So where the positive and negative amino acids are essentially equal to each other, that is where the maximum unfolding is going to happen. And so this was quite surprising. Not only that, but we also found that these conductive style events do not depend on Cas9 charge, which was previously thought of in the field, that they would yield this different charge dependent properties. So yeah, I'll skip that. So most of the work on pressure was done by Vinay Sharma, who's now a professor at IIT Jammu. And my current postdoc did a bit of the Cas9 work, Nuon. Also, my graduate students, Lauren and Nassim, did a bunch of work with the DNA as well. And my funding for this was Human Frontier Science Program. So if there's any questions, I'll be happy to answer them. Thank you.
Thank you so much, Kevin. Really amazing talk and so many interesting things and questions that I think all of us would like to ask you. And perhaps I will start breaking the ice. So I think this is a fantastic tool to characterize complex interaction in biological samples and liquid. But how do you go about approaching a liquid where the composition is not known? So for example, if you want to analyze a biosample with multiple proteins, RNA or bacteria, how would you go and bring this apart?
Yeah, so you got right to probably the limitation to nanopores is that, you know, most experiments are done with a purified sample. When you go into a complex heterogeneous sample, it really becomes almost impossible. What we do in the lab is we do it step by step. So if we have maybe say 3 different molecules that we're trying to look at, we would run each of them individually and then mix them together. Other than that, you know, it's really hard to untie what molecule is going through the pore. So yeah, we have to do it one by one.
And I completely understand, and that's would be a follow-up question. So how do you go about sample preparation? Do you purify the sample? Do you try to obtain a certain concentration? So what kind of preparation do you have to do?
So we assume that the sample that we order or is given to us by a collaborator is pure enough where I would say if there is a contaminant, that it would be at a much more diluted state than the target. So if that's the case, as long as we can discriminate So for example, say if we had nanoliposomes that are about 100 nanometers, and there was maybe a bacterial cell that got mixed in, usually, for sure we can discriminate between those two different things. And so if 99% of the events correspond to the smaller entity, then we can just kind of throw away the outlier. And so either throwing away the outlier is done, or sometimes we have to go back and re-purify the sample. But yeah, we basically have to have it majority of the events coming from the thing that we want it to come from.
Okay, I understand. Thank you so much. And I think I'm starting to get a big picture of it. And perhaps of all, we could use it ourselves in the future, perhaps with your help. And so I'd like to ask anyone else to ask questions. Trying to see if there is anyone raising their hand, but I didn't see anyone. So feel free, if I didn't see you, to unmute yourself, please, and go ahead. Well, in the while, perhaps I'll go and ask you another question. So do you think there is potential for nanopore technology for biomedical diagnostic of infectious diseases? So finding out if someone, for example, has COVID or other things where you have a trace of RNA or something like that.
So diagnostic sensing of RNA?
Yeah.
So like you probably have figured out that the nanopores that we use in our lab cannot get sequence information. Mm-hmm. So the solid state nanopores so far have not been able to achieve DNA sequencing, but we are able to get molecular properties. And so as far as RNA sequencing, I don't think that's going to be possible for a while. However, we can look at RNA folding. We can look at how RNA binds to different other molecules, either a protein or DNA, and look at those interactions. So similar to the Cas9 work, if there was an RNA molecule that is complementary, so if this is single-stranded DNA, and we have a complementary RNA, we should be able to detect, this RNA being present on the DNA. And so doing things like that is definitely possible. Whether or not that has any diagnostic meaning, I'm not sure, but it's up to, we can design it so that it does, essentially.
Very good direction, actually. I see Omi does turn on his camera. Omi, do you have a question?
I just want to thank Kevin. That was a great talk. Just because I was interested in the machine learning aspect of the work that you mentioned. And I just want to know if you have these data available anywhere, or this data is publicly available or not?
The machine learning like program?
Yep. No, the data itself.
Oh, I can share it with you. Okay. This work is going to be published probably pretty soon. But yeah, if you're interested in looking at it, or yeah, I can share the code that I use to do the classification as well.
Yeah, that would be interesting, because I just want to know from the bioinformatics aspect of it, what kind of work can be done to expand. Yep.
Kevin, I have one question. Is it possible to measure multiple sequence-dependent properties during the process of translocation of any biopolymer?
So if you have a biopolymer and there's some different sequence there?
Yeah, like multiple sequence properties.
So if it's different enough that you're able to, for example, if this is single-stranded DNA, you can hybridize a probe to it that binds when the sequence is present and then doesn't bind when it's not present. And so that could work to recognize certain domains. So if you have a complementary strand that binds to the DNA, you can even make this a bit bigger by adding some particle. And so as it goes through the pore, whether this DNA, this probe and particle is present will definitely be detected by the nanopore.
Thank you.
Kevin, if I still have time for a question, but first, I would like to thank you for the amazing work that you're doing. It's quite exciting. And I think it's a technology that, with a bit more maturity, is going to definitely open a lot of new avenues. And we would be really excited actually to see if there is any way to do things there. But one question that actually follows up a bit with what Antonio was asking. And you mentioned that so far, synthetic nanopores, they cannot do sequencing. So what is the main reason? Because you mentioned that actually you have the possibility to really tailor the pore with the size exactly that you want. So what is the main limitation then between the natural nanopores and the synthetic ones?
Yeah, good question. Yeah, there's two main things. One is the speed at which DNA travels. And the other thing is at least what I call the Kamer issue. And so this can be taken care of. It's typically taken care of in the biological pore by having the alpha hemolysin and then putting essentially a motor protein that kind of ratchets the DNA at a controlled speed. So that's how Oxford Nanopore takes care of the speed issue. However, Oxford Nanopore still has this issue, which is essentially that there's multiple bases inside the sensor at a certain time. And so for Oxford, this is about 8 bases. So there's 8 bases that are contributing to the current signature. And so we're actually getting a moving average of the DNA bases. Not one base means this. It's actually a chunk of bases that give us a signal. And so this is even bigger of a problem for solid state nanopores because the membrane tends to be a bit thicker. And that's where 2D materials come in. So using a graphene, for example, which is about 0.3 nanometers, this should be about the distance of the base. So we should be able to use 2D materials to reduce this Kamer issue. But like I said, even if you do that, there's no way to get this motor protein on graphene. So it's really hard to get rid of this issue plus this issue. So yeah, those are the two main issues here.
Thank you very much.
Yeah.
So we have time for a last question from the audience, if there is anyone. Okay, else I would like really to thank again Kevin for this fantastic talk. Actually, it was very interesting, and I hope we can eventually follow up with some collaboration with our school. And also, if you plan to visit Australia, Sydney is a very beautiful place. Very happy to host you here and show you our school and their colleagues. So thank you again, Kevin.
Thank you so much.
And thank you everyone for joining today. Bye.
Bye.